I'm delighted, if not slightly nervous, to be speaking here today. And I'm going to talk to you about two really large national genetic data collections, which I believe are going to have a huge impact and have huge possibilities for research in Lynch syndrome. And so they are the National Clinical Genetic Testing Dataset and the English National Lynch Syndrome Registry. So I'm going to start off by talking about how those datasets were formed before comparing the two, and then moving on to talk about just a few of the possible uses for those datasets that have been completed, that are ongoing, and that would be possible in the future. Okay. So the National Clinical Genetic Testing Dataset is a national collection of NHS clinical genetic testing results, unified into a common data structure. And that's the really important bit, is because bringing it together, putting it in one data structure is what allows you to be able to do research using this data. Now, this is a huge and long-running project. I think it was first sort of mooted in 2015 and involved all of the genetic laboratories that have historically done testing for Lynch syndrome in the UK, submitting their data to the National Disease Registration Service. They also submit other data, BRCA data, and data on other cancer susceptibility gene syndromes as well. This was a very challenging project, and these are a few of the challenges that they faced. First was how those genetic laboratories submitted their data, and there are a variety of ways that they submitted it, from the cleanest at the top being a single item of information in a single box, to the dirtiest being a word soup at the bottom there, where they just send what was written on the report. And so the challenge for the NDRS team was writing bespoke scripts, computer coding scripts, for each lab to extract that data from that word soup at the bottom and put it into a nice clean format for researchers to be able to use. Another difficulty that the labs faced in collecting all of this information to submit is that sometimes these records were from the late 1990s, the early 2000s. They may have been on old computer systems filed in the back room, or maybe just completely inaccessible. So for that reason, some of the labs weren't able to submit some of those really early records. And by comparing the data that we do have to the data we expected to have from historical audit data, we reckon that between 96 and 2020, we've got about 60% of those testing records for Lynch syndrome. And then, as with any project that uses real-world data, you will encounter human errors. For example, typos, an extra O here, an N for a U there, and this does need cleaning and does affect what you can do with that data. Okay, but that aside, as a complete data set, we can make some observations. This is a fairly obvious observation, but over time, the amount of testing for Lynch syndrome reassuringly has gone up. Over time, how we test for Lynch syndrome has changed as well. In the early 2000s, tests were mainly just for those two genes, MLH1 and MSH2, because the other genes that we now test for, MSH6 and PMS2, were discovered later and also slightly more difficult to analyze in the lab. But testing for the four genes kind of took over from 2007 onwards. Over that same time period, the proportion of tests that turn out to be positive and do diagnose Lynch syndrome has remained largely the same. And for a new person coming from a new family to be assessed for Lynch syndrome, we call them probands, the rate of making a diagnosis in Lynch syndrome for them is about 15%. Okay, so that was the National Clinical Genetic Testing Dataset. And just to reiterate, the submitter of that data is the genetic laboratories, the people that do the tests. And the data itself is the test results, both positive and negative results, so where it did find Lynch or didn't. Now, as I mentioned, because of the difficulty submitting that really early data, we think that this data up to 2020 was only about 60% complete. It was wondered whether we could form a Lynch registry, so a register of all patients diagnosed with Lynch syndrome off of that data, but because some of it is missing, that wasn't possible, and so a second project had to be formed to do that. And so now I'm going to talk about the formation of the English National Lynch Syndrome Registry. Oops. So for this, you needed to, at the time when this project was started, go back and ask the clinical genetic services this time, so the doctors, the counsellors, the administrative staff in the services, to go back and identify everyone they had diagnosed with Lynch syndrome and submit those lists to the National Disease Registration Service. I mean, it sounds very simple, but you cannot overemphasize what an absolutely colossal amount of work that was for the clinical genetic services. And then in order to make sure that we're capturing everyone from this point onwards, I'm going to start calling them the NDRS because it's just too much of a mouthful. The NDRS had to make a submission form so that all Lynch syndrome patients diagnosed from that point onwards could be submitted at the point of diagnosis using a real-time data collection encrypted to NHS standards so that they can be automatically pulled into the Lynch syndrome registry. And the nice thing about that is, because built into that portal is all the data cleaning steps, so no additional data cleaning is necessary. Obviously, those old records needed quite a bit of validating and data cleaning before being pulled into the Lynch registry. So pulling everything together, as of July last year, there were 9,030 patients in the Lynch syndrome registry. And the main use for this at the moment is to enroll people into the National Bowel Screening Programme, which will be a huge patient benefit to the organisation of bowel screening. I just wanted to pause here. I mean, Adam slightly stole my thunder with my salient and sobering point here, but if we... 9,030 unique patients as of July 2023. This is the first time we've actually been able to say how many people are diagnosed with Lynch syndrome in England. Which it seems like quite a simple number, but we've never been able to put a figure on it before. If we take the population of England to be about 56 million, and the frequency of Lynch syndrome, I'm using a slightly different figure, but the frequency of Lynch syndrome to be about 1 in 279, we would think there'd be about 200,000 people in the UK with Lynch syndrome. So that figure of 9,000 people currently diagnosed is a sobering less than 5% of people who have Lynch syndrome who currently know about it. Okay. So with this registry, we can say a few things about the people that currently have a diagnosis of Lynch syndrome. And they may not, and they probably aren't representative of the whole population of people who have Lynch syndrome. We know that there's slightly more females than males who currently have a diagnosis, and that there are more in the older age brackets than the younger age brackets, that there are more from less socioeconomically deprived backgrounds than more socioeconomically deprived backgrounds, and that the gene most commonly identified as the pathogenic gene is MSH2. And people who currently are diagnosed with Lynch syndrome are not evenly distributed around England. Okay, so just to reiterate again and to contrast it against the genetic testing data set, the Lynch syndrome registry, that data came from the clinical services, and the data is diagnosed Lynch syndrome patients. Now, there is some overlap between these two data sets, but we can link the two data sets, and they can help with the completeness of those data sets. For example, for 1 in 4 people in the Lynch registry, we don't know when they were diagnosed. And for researchers, that's actually quite important information to know when someone started screening or started knowing that they had Lynch syndrome. But you can top that information up from the first data set, from the data set of test reports. And similarly, it's useful to know what specific DNA change a person with Lynch syndrome has. And again, that data can be topped up from the genetic testing data set. Okay, so that's enough about that, and move on to talk about some of the uses of this data. So one of the uses for the genetic testing data set, that data set of test reports, is for variant interpretation. So when a new person goes forward to be tested for Lynch syndrome, their blood arrives in the lab, they sequence the blood, and they find a DNA change. What a clinical genetic scientist has to do, especially if that DNA change has not been seen before, is they have to assess a number of different pieces of information to be able to say whether they think that that DNA change causes Lynch syndrome or whether that DNA change is just harmless noise, it doesn't mean anything. And one of the pieces of information that can be used to make that call is population data. And so something that we can get from the genetic testing data set is we can ask, okay, for this specific variant, so in this case, MSH2 1862G to T, How many times do we see it in people who've come forward for Lynch syndrome testing in England? In this case, 7 out of nearly 10,000 people. And we can compare that to how often we see it in the general public. In this case, 0 times out of 51,000 people. And that is a piece of the puzzle that helps scientists say whether someone's DNA change does cause Lynch syndrome or not. And so ultimately, publishing this information, which is done on this website, Canva UK, which is used by UK laboratories and international laboratories, ultimately it helps more people arrive at a diagnosis. Okay, so I should have said both of these data sets are held in NDRS. And the benefit of that is that the NDRS holds a wealth of other data sets as well, probably most famously the National Cancer Registry. And so the fact that that it's housed in the same place means you can link these data sets to the Cancer Registry, which vastly increases the applications of these data sets for research. So this has already been touched on, but I'll go through it again to make Adam Shaw's job a lot easier in terms of auditing how well NHS services are sticking to those nice guidelines of putting all bowel cancers through a series of tumour tests for Lynch syndrome, followed by a blood test, a confirmatory blood test for Lynch syndrome, can be audited using these data sets. So if we take the National Cancer Registration data set to isolate everyone who got bowel cancer in 2019, we can see how many of them are progressing through that pathway as they should to be assessed for Lynch syndrome. How many are receiving the tumour testing can be got from linking this data set to the molecular pathology data set, and how many then go forward if it's necessary to have genetic blood testing for Lynch syndrome. And doing this, what we see is that this is 2019, so it will be a lot better now. Not everyone was even entering the pathway. And that of the people entering the pathway, some people were stopping at steps and not progressing through to the next step. And if we replicate this analysis in endometrial cancer, we see exactly the same thing, with attrition at each step, with people falling off that pathway and not making it all the way through that testing pathway. And in its simplest, what this means is there are missed opportunities to diagnose Lynch syndrome and underdiagnosis of Lynch syndrome. Again, this is 2019, so it's not quite fair because the guideline that says you should do this didn't come in until 2020. But the point stands. Okay, so now moving on to talk about the long-term study of people with Lynch syndrome, cohort studies. If we take everyone in the Lynch syndrome registry and link it to the National Cancer Registry, we can characterise in great detail the cancers that people with Lynch syndrome get. And I mean, this is not huge news, but it's reassuring that it replicates what's been demonstrated before, which is that if we take people who didn't have cancer at the time their Lynch syndrome diagnosis was made, and we follow them forwards in time, those that do develop cancer do so at a younger age, 10 to 20 years younger than in the general population. So those patients with Lynch syndrome here are in turquoise and the general population is in orange. Sticking with that group of people who didn't have cancer at the time they were diagnosed with Lynch syndrome, if they do go on to develop cancer, they tend to be diagnosed at earlier, and usually that means more treatable stages, which is reassuring for why it's important to have a Lynch syndrome diagnosis, why it's important to be aware, be symptom aware, and to receive screening. It really does translate into cancers being diagnosed at earlier, more treatable stages. Okay. Taking that linked data set and linking it again to information on what surgery, what chemotherapy, what radiotherapy patients with Lynch syndrome receive should they get cancer, allows us to address questions which until now haven't really been studied in great detail. What's the best treatment strategy for bowel cancer in patients with Lynch syndrome? After a bowel cancer, what's the chance of getting a second cancer? And these are two questions that I look at in my research. If I come next year, I hope to have something to tell you about it, but I'm in the thick of it at the moment. And we can also look at questions like, how does colonoscopy change cancer incidence, stage, and survival in Lynch syndrome? And there are 100 other questions that you could ask with this data. Okay. And then lastly, I wanted to touch on some things that could be possible now that we have these data sets. Okay. Should we go about historic case finding? So this is the most recent testing criteria for, well, it's a snippet of it, for Lynch syndrome. So if you have 2 Lynch-related cancers, so if we look at number C, 2 Lynch-related cancers, or B, any Lynch-related cancer under the age of 50, then you could be considered to have tumour testing, so testing on the tumour for any features of Lynch syndrome. Now, there will be patients who 5, 10, 15 years ago would have met these criteria, but they weren't eligible at the time. Had they been diagnosed today, they would be eligible for this testing. Because we have the National Cancer Registry and we have a data set which tells us which people have and haven't been tested, we could quantify these patients. We could find these patients. We could retrospectively go back and offer them this testing. And this is one thing that these data sets could allow us to do. But the other thing that it allows us to do is to answer questions like, well, if we were to do this, how many patients would that be? How many Lynch syndrome diagnoses would we make? And if you extend that questioning and modelling further, ultimately how many cancers we could prevent, which in an NHS which has a finite budget, you need to make that case for any project that you want to do. Okay, so I'm coming to the end now. These 2 data sets, I believe, can have a huge, I've just gone through a few, but have a huge number of potential applications across the whole journey of being diagnosed with Lynch syndrome and for the rest of life. From one, helping more people get diagnosed with Lynch syndrome in the first place by contributing to variant interpretation. 2, studying people over a long period of time. This evidence from that research will ultimately help guide for those people that are diagnosed, how should we screen them, what preventative measures should we take, and should they develop cancer, what treatments are best. And then once you have all this evidence, you really need to have those tools to be able to make sure that the NHS is implementing that evidence. And so having these data sets allows us to audit what NHS services are doing. So that's me. Thank you very much. This work is, of course, the work of a huge number of people, and I hope that I've captured them all on this slide.